Hey everybody, we are looking at chapter 8 now. So we're moving on from chapter 7, where we're learning all about the immune system, its structure, and the cells that function within it. We're moving on now to look at what it actually means to be immune to something. So focusing on the action of those B memory cells, um, how that is seen in larger populations, in herd immunity. And some deficiencies of the immune system, with 3 specific examples that we focus on there. So we'll start out looking at how, or the different ways that we can become immune to certain pathogens, comparing some key terms being active and passive, and natural and artificial, what they mean. Vaccinations, how they work, what a vaccination program means, and how herd immunity can be obtained in a population and why that is a benefit. We're seeing that in real life, of course, at the moment with the big call out to be vaccinated against COVID-19 and looking at achieving a form of herd immunity in our population. And then the deficiencies of the immune system, we'll look generally at what autoimmune diseases are, but specifically at what it means, or what multiple sclerosis entails, as an example. Immunodeficiency, the example that we focus on is HIV, and allergic reactions, with the example of being allergic to pollen or suffering hay fever, which a few people are probably familiar with. And then we wrap it up by looking at the use of monoclonal antibodies. So antibodies being a molecule that we're now familiar with, and how we are using that to as a benefit to our species in combating the terrible disease cancer. Right, so that is the scope of chapter 8. And it is a lot smaller a chapter than chapter 7. So let's see how we go. Okay, so talking about acquiring immunity. There are 4 different ways that you can come across immunity, that you can become immune to something. So there are 2 broad forms of immunity. You can have long-term immunity, where your body has a memory and is able to respond to a pathogen in the future, or short-term immunity, where your body can get rid of a current infection, but you won't retain any memory of that immunity in the future. So to define the words first, active immunity is where your body, or your own immune response has responded to an infection. So where your immune system has done the work. Passive immunity is where antibodies, the result of your immune system, have just been given to you. Your body hasn't actually manufactured them. Natural immunity is where you have encountered your immunity under natural circumstances, real world circumstances. Artificial immunity is where you have come across your immunity under clinical circumstances. Has been artificially administered. So what do they look like when you combine those 2 words? A natural active immunity is where, in real world circumstances, outside of a clinic, outside of a doctor's office, you have encountered a pathogen and your body has actively fought against it. So this is any normal infection. You have been infected with rhinovirus and suffered a cold. You went to a friend's birthday party and they had the chicken pox, and then you get the chicken pox, full blown itchy sores and everything. So that is a natural active immunity. Because your body undergoes an immune response and um, you produce antibodies and B memory cells, that is a long-term immunity. Natural passive, oh, actually, I'll do it the other way. Active artificial, or artificial active. Active artificial immunity is where your body has done the work, you've gotten rid of the pathogen, but you've been exposed to that pathogen under artificial circumstances. This is vaccinations, immunizations, which I'll talk about in a better detail in a minute. But where your body has undergone its own immune response, so still giving you B memory cells and long-term immunity, but you were exposed to that pathogen artificially in clinical circumstances through an injection, through a vaccination. Okay, passive immunity, the two examples will be where you have been given antibodies. So you can be given antibodies naturally, and the two examples of that are antibodies that cross the placental barrier. So while a mother is pregnant, there are some antibodies that can cross the placenta and provide that short-term immunity to the baby, and also through breastfeeding. So both maternal examples and both ways that you can naturally acquire passive immunity. The antibodies in question, which ones of these can cross? So IgG antibodies can cross the placenta, and IgA antibodies are found in secretions such as breast milk. So those two would be examples of antibodies that are given passively from mother to child. The last one then is a artificial passive immunity. So how can we acquire antibodies under passive circumstances, under clinical circumstances? Used as an emergency treatment against infection. So the best examples we have are antivenoms. So where someone has been bitten by a snake, we have antibodies that can be administered that will attach to that venom, and it can be gotten rid of out of the system, but they're administered by an injection. So antibodies would be an example, antivenom would be an example of an artificial passive immunity. Another one would be treatment against or treatment after exposure to tetanus, where antibodies can be administered and that tetanus bacteria can be targeted and eliminated from the body. So they're four key terms, and being able to use them in conjunction with each other and explain what that would mean in terms of how the immunity has been required is a big part. All right, so starting out looking at what vaccinations are, what the term immunisation, so what immunisations consist of. Lots of talk in the news about vaccinations recently, the benefits and activity of them. So for a basic representation, vaccines consist of an attenuated or weakened form of the virus. So what we're achieving is presenting the antigen that is carried on the pathogen of interest, presenting that to your immune system without your body actually falling ill. So without being subjected to the infection itself, you are giving your immune system a a wanted poster. You're giving them a snapshot of what the antigen looks like so that they can look out for it should you come across it under natural circumstances. So after the recipient is exposed to the antigen, your immune system will launch its response as per usual. The steps of an immune response to a vaccination are exactly the same as what we've covered when we've talked about the adaptive immune response. The antigen will be recognized as non-self. Second line defenses like macrophages and dendritic cells will take the antigen back to the lymph node, present it to the T helper cell, which will initiate the differentiation of B lymphocytes, producing their mass amounts of antibody. And again, we've seen this graph representing a vaccination in this case, but we talked about it being an infection. So this can represent an initial infection, so being exposed to the disease itself, or a vaccination. This marks the time that your body is shown the antigen, that the antigen comes into contact with your body. This is your first immune response. So it takes a number of days to build and produce enough antibodies to get rid of that antigen that is infecting your body, or the vaccination in this case that has come into your body. Your antibodies can then deplete over time, although you do remain low levels of antibodies still circulating in the lymphatic system. And then after an unknown period of time, so after your first exposure, when you are exposed to that same antigen a second time around, your body builds that immune response that much faster because of the presence of your memory cells stored in your lymphatic system. So if this was the point of vaccination, you still get that initial immune response. Then, should you come in contact with the pathogen itself, your immune system can get rid of the pathogen before you even know you're sick. So memory cells provide that rapid response upon second exposure. All right, so what is the immunization schedule? You become very familiar with this when you become a parent. It's something that all my kids have gone through, and you yourself probably went through as a child. So it is the number and order of vaccinations that can be given at a certain age. So you can see the very first vaccination that babies receive at birth is against hepatitis B, and it's given within 7 days of birth. And that's just the name of the vaccine brand that is used. So you've heard the words AstraZeneca and Pfizer. They're the brand names of the COVID-19 vaccine. These are all brand names of various vaccines that are given during a child's development. So all the way from birth through to recently, enough for you guys, year 10 at secondary school, where you would have received your diphtheria, tetanus, and pertussis vaccine being the last in your immunisation schedule. So certain vaccines can only be given at particular ages. It's only safe to administer them once a baby has reached a particular milestone. And some vaccines require 2 to 3 doses to be effective. So probably, what can we pick out? This RotaTeq vaccine against rotavirus, from memory is gastro, I think, a gastro-inducing virus. That's just my memory of it from when the kids received it. So RotaTeq is given at 2 months, 4 months, and then again at 6 months. So that one requires 3 doses to be effective. I'll go back one just to talk about why they need that booster shot. So you can see after secondary and even tertiary exposures to an antigen, your immune response builds every time. And importantly, you can see that it takes longer for those antibodies to disappear from the body. So you're retaining a stronger immunity for a longer amount of time with every booster shot that is provided. Importantly, there is no one answer. All immunizations are different. Some of them are oral, some are injected, some require two courses to get full strength immunity. Some will have three, some only need to be taken once. Some you need to receive every year, like the flu shot. So every vaccine is different, and we've seen that developing in front of our eyes with the development of the various vaccines against COVID-19. They work in different ways. They have different components as part of their makeup. Um, they they require different doses. They're not all the same. They are basically achieving the same goal. They are presenting your body with the antigen, but how they go about that, there's lots of different ways that they can do that. So that's a few examples. Right, key term that comes up in chapter 8 that is a simple idea, but you want to make sure that you understand it well, is this idea of herd immunity. Definitely something that we've heard about firsthand in the news at the moment. So looking to achieve herd immunity in a population. I would like you to do this little activity as part of your OneNote task. So choose a disease that you have been vaccinated against and find out the following. So the immunisation schedule on the previous slide will be helpful there. What age can you be given the vaccine? When was the vaccine created? Has there been a decline in the number of cases? Look at the statistics. And how does herd immunity contribute to the decline in cases? The idea of herd immunity is represented pretty well in this diagram. So you've got three classes of people. People that are not immunised and otherwise healthy, people who are immunised and healthy, and people who are not immunised and they are sick or contagious. If we've got a population where nobody is immunised, if you end up with one or two people in that population that have the disease, it will spread rapidly to other members of the population. If we have a population where some people are immunised, again, you're going to have pretty rapid spread of the disease when you have one or two people infected. If you have a population where most, and there's a stat like 90% herd immunity is ideal, where most people in the population are immunised, even though we've got these few people that aren't immunised, it's very difficult for that disease to be passed on to those unimmunised people. Why are these people not immunised? So why isn't herd immunity such an important idea? Because we have people in our population who can't be immunised. Sure, we have people who choose not to be immunised. The campaign for anti-vaccination crops up from time to time in the media, and there's a OneNote task around the ideas that are presented by that group, and asking you to do a little bit of research and find a rebuttal, if you like, against the ideas that come out of that campaign. So there are people who choose their own values to not be immunised, but there are people who cannot be immunised. The babies that are too young to have received their vaccinations. People who are their health is otherwise compromised. They may not have a functional immune system that can cope with a vaccination and provide them with any kind of worthwhile immunity. So herd immunity is important to protect those people in our population. If we are healthy, otherwise healthy and can be immunised, you should be immunised. Herd immunity works to protect people in our population who cannot be immunised. Now, that link takes us to one of your OneNote tasks, which is this website here. However, when I'm trying to use it tonight, it is not working very well. It should be an interactive map of immunisation rates across Australia. If when you click the link it works well, then use it. There are some statistics already given down the bottom that you might be able to make use of, but we'll see how you go. I'm not sure why the map's not working tonight. Okay, so then that takes us to our deficiencies or diseases of the immune system to have a look at. Okay. So first, we'll look at autoimmune diseases. In general, autoimmune disease, I like to pull apart a word. You know I like to pull apart a word to find out its meaning. Autoimmune. Auto, when you see the word auto in bio, it means self, autotroph, self-energy, self-food. Autoimmune, self-immunity. It is where the body has had a failure of the generation of self-tolerance. So I talked about that as part of our discussions on the adaptive immune system. When your B and T cells start developing, they undergo this process of generation of self-tolerance, which means that antibodies, or if antibodies are produced in your body that recognize self antigens, they are destroyed, they are gotten rid of. Unfortunately, if someone is suffering an autoimmune disease, that means that they are capable of producing and do produce antibodies that recognize their own antigens, their own body's antigens, as being non-self. And normally it's a particular tissue where the antigens present on that particular tissue are recognized as non-self. In multiple sclerosis, the target tissue is the myelin sheath surrounding the axon of the neurons. In your OneNote tasks, I have linked each one of these diseases of the immune system. Has a bit of an explanatory video to go along with it. So watch those. That'll help you understand multiple sclerosis and the action of the immune system when we are suffering multiple sclerosis. But the target tissue being the myelin sheath, we know the structure of an axon, a neuron. We know that myelin sheath is present on an axon to do two things: protect the axon and to help the traveling of the signal, the action potential along that axon. When there's sufficient myelin sheath, the electrical signal will travel down the axon of the neuron well. Once myelin sheath starts to be eaten away, taken away, the message will perhaps fail, won't pass through the neuron, or pass very slowly along the neuron, which helps to then explain the symptoms of multiple sclerosis, where people lose functionality of their nervous system, can't move their muscles because the neurons won't communicate to them anymore. Okay, so that was multiple sclerosis. Immunodeficiency. It's likely that you're going to mix up the words autoimmune and immunodeficiency. So really try to analyze the words. Autoimmune, self-immunity. Immunodeficiency, deficient immunity. So it's where the immune system is impaired or fails to respond in a normal way. That's the general explanation. The example that you need to know, as I've already mentioned in class, is HIV. This is quite a good video and will explain pretty well how HIV works with some good graphics. But HIV virus, which is the precursor virus to AIDS, AIDS standing for acquired immunodeficiency. HIV leads to AIDS. So AIDS is where we've got a completely defunct immune system. HIV is the virus attacking the immune system that will eventually get us to the point where the immune system's not functional. HIV, the virus, works by infecting, obviously, your own body cells, and a particular cell being the helper T cell. What effect would this have on the immune response? Let me go back to one of our previous PowerPoints, just as a quick reminder. That's where our T helper cell was discussed. So remember, our T helper cell is the first point of call for the antigen presenting cell. T helper cell will recognize the antigen being presented and then facilitate the binding to the correct B lymphocyte. It will signal that B lymphocyte to undergo clonal expansion and will result in the antibody being produced. So if we have a virus infect our T helper cells, it's a sad fact that once a virus infects a cell, that cell will eventually be destroyed. So the virus will inject its genetic material into the T helper cell. The T helper cell will produce all the parts of the virus that are required to build another virus. Once there's enough virus present in the T helper cell, the cell will be killed and the virus will be released into the body. So if we're eliminating a good supply of our T helper cells, you can draw the link to what effect that's going to have on the immune system. If there's no T helper cells, B lymphocytes can't be stimulated or the humoral response can't be initiated. So B lymphocytes will not be able to produce B plasma cells and B memory cells. And that is the whole adaptive immune response. So people who are suffering with HIV will eventually get to a point without treatment where they don't have functional T helper cells. And so therefore their adaptive immune response is shut down. It is not HIV that will kill the patient. It's their inability to then fight off another disease that will infect their body. So something that as simple as a common cold that your body could get rid of easily enough with help of your adaptive immune system. Somebody with HIV unfortunately doesn't have that ability. Okay, and then our last disease of the immune system to cover is hypersensitivity. There's a lot more detail given in the textbook about different kinds of hypersensitivity. Definitely have a read through them. There's some textbook questions about them. I'm just going to talk to the basic idea of hypersensitivity and specifically what a pollen allergy would look like. So hypersensitivity is triggered when the body produces IgE antibodies upon exposure to an allergen. Now, we talked about the different kinds of antibody. And said that there's different shapes to antibodies that we can get, IgE being one of them. But there were others that you were asked to have a look at. So if you can remember, I can go back to our textbook. We've got these classes of antibody and they vary, some of them in their shapes. But their constant region is the same within their class, but there's these 5 varied shapes of antibody. So normal antibodies, the antibody that is produced mostly in your body is our IgG antibodies. What does it say? most circulating antibodies, more than 80% is the estimate, would be IgG. So that's our regular run-of-the-mill antibody. But IgE down here, they're a slightly different class of antibody, and they are unfortunately the ones involved in allergic reactions. So the way it goes is, if you suffer from hay fever, when your body was exposed to that allergen, the shape of the pollen when it first entered your body, when your body underwent the immune response to produce antibodies, unfortunately, it was a B lymphocyte that had an IgE antibody attached to it that responded. So now that means that IgE antibodies are released in your body every time you're exposed to this allergen, which is fine, though, and it's fine on the first exposure because those antibodies work pretty much the same. They will attach to the pollen and it will be eliminated from the body, digested by phagocytes, normal run-of-the-mill response. However, the difference between an IgG and an IgE antibody is that IgE antibodies, their constant region has a shape that binds to the surface of mast cells. So this area, this base area of the IgE antibody, it has the capacity to stick onto mast cells. With the problem being then that when these antibodies get triggered again, they have an effect on the mast cell which causes an overreaction, a bigger release of histamine the second time around. So it's not the first exposure that will do that will cause the hypersensitivity or that will give you the symptoms. It's the second exposure to the pollen. So the first time around, we produce these IgE antibodies. The IgE antibodies stick to the surface of the mast cells. The second time we have that pollen enter the system, and I think this is important, so I'll just point it out. The pollen needs to bind to two recognition sites, so to two separate antibodies for this to have an effect. So once the pollen is bound to two antibodies on the mast cell is triggered to produce lots of histamine. What does histamine do? It induces inflammation. And so where you're feeling the effects of hay fever in the eyes and in the nose and in the throat, that's where those pollen spores have been taken in and are having an effect on the mast cells in that area, causing the inflammation there. So inflammation response is the same as it normally would be. The increased permeability of the capillaries of the blood vessels and the increased blood flow causing that itchy, swollen sensation. Okay, so what's the treatment? We have brought that up before, but if we've got histamine being released, which is triggering the inflammation response, antihistamine is a molecule that can neutralize that histamine signaling molecule and cut it off there at that point. But another treatment would also be something called microexposures. Microexposures, if you know someone who's ever suffered greatly from hay fever, or you yourself perhaps, microexposures is a treatment that some people turn to, where your body is given very small amounts of the allergen, the thing that you're allergic to. So if it's pollen, you're exposed to small amounts of pollen at a time, microexposure. The hope is that instead of the pollen attaching to these already existing IgE antibodies, or attaching or triggering the memory cells that would exist already that produce those IgE antibodies. The hope is that we can get an immune response from a B lymphocyte that has an IgG antibody. Because if you can stimulate an IgG antibody and produce memory cells that have IgG antibodies instead, then we're not going to get that immune response happening anymore, or it will be in a very reduced amount because you've got other memory cells that can produce different antibodies. All right, so have a watch of that video and hopefully we can understand hypersensitivity. All right, the very last one is monoclonal antibodies. So firstly, what monoclonal antibodies are, and then we'll talk about a couple of different kinds of mAbs that we are using pretty well and successfully in the fight against cancer. But monoclonal antibodies, it's a very ingenious idea. We've got a case, so we're now familiar with this beautiful little molecule that has the ability to recognize particular cells in the body, to recognize particular antigens. So using that to our advantage, can we get these antibodies to target specific cancerous cells? So the first thing that has to happen is the production of the monoclonal antibody. So monoclonal just means that it's being cloned from one source, and they are produced, or it's pretty well depicted in this diagram, the process of production. So the antigen in question, whatever we want the monoclonal antibody to be specific for, the antigen is injected into an animal host, in this case a mouse. Once the immune response has been triggered in the host, the antibody forming cells will be isolated. What are the antibody forming cells? The B lymphocytes that have been activated. So the B lymphocytes that were activated must be the ones that carry the antibody that is complementary to the antigen in question. And then this is a much more complicated process than's being presented here, but it's safe for you to just recognize that this fusion process takes place. So the B cell is fused with what we call a myeloma cell to produce something called a hybridoma. The hybridoma is meant to be, they call it an eternal cell. They call it it's a cell that has a longer shelf life. The B lymphocytes themselves will die off after a certain period of time. They live longer than a regular B lymphocyte, so allowing them to have a longer lifespan. Those hybridomas get cloned, and they all have the capacity, the ability to produce an antibody that we know is complementary to the original antigen. If we're producing this antibody in response to a cancerous growth, the antigen that's present on the surface of the cancer cell will be what was injected in the first place in order to form those antibodies. Okay, so that's nice. We've got an antibody that can attach to specifically that cancerous cell that we want to get rid of. A couple of ways that we can manipulate this monoclonal antibody now that makes it even more usable. So conjugated mAbs. Conjugated mAbs are ones that have a radioactive isotope attached so that it can be delivered directly to the cancerous cell. So working a lot like chemotherapy would anyway, but in a very specific manner. Chemotherapy is administered to the body to kill off cancerous cells, but it's given to the whole body. A conjugated mAb would if we've got the radioactive isotope attached on one side and the other side is able to attach to the cancerous cell, you're delivering that radioactive isotope directly to the cancerous cell. So it is a form of specific targeted chemotherapy. Bispecific MABs, they are ones that have the ability to attach to the cancerous cell and to cytotoxic T cells. Cytotoxic T cells, we know, are the ones that can detect cancerous growth and signal to that cell to get it to undergo apoptosis. But they have to be able to find the cancerous cell first. So bringing the cytotoxic T cell directly to the cancerous cell using a bispecific MAB, beautiful little situation. Okay, so for chapter 8, you've got 3 separate OneNote tasks to have a work through. They are much shorter than the chapter 7 ones, so they shouldn't take us very long at all. And if you've watched this video before class, I'll be able to help you out with these little tasks in class. Good luck, guys. Talk to you soon.